What drives us to eat, and how does the body maintain energy balance? Hunger is the physiological drive to eat, which may cause gastric discomfort stemming from contractions of the stomach when it's empty. This is not to be confused with appetite, which is the psychological desire to eat. Appetite can be influenced by smell, taste, or thought of food, and also tells us when we think we should eat even if we aren't hungry at these meal times. Hunger and appetite can coexist, with hunger driving thoughts about food intake. Once hunger has been appeased after eating and we begin to feel satisfied, the body reaches satiation and hopefully eating stops at this point. The resulting period of fullness for the next several hours is called satiety. These feelings of wanting to eat and being full are driven by activity in the brain. The brain, especially the hypothalamus, is the main regulator of the body systems that start and stop feeding in order to meet energy homeostasis. There are specific areas within the hypothalamus that trigger food intake or feelings of fullness in response to stimulation from hormones, peptides, and neurotransmitters. The two key hormones most integrated in energy homeostasis processes are ghrelin and leptin. Ghrelin is a hormone secreted in the stomach and reaches its highest levels right before a meal, signaling to the brain that hunger is present and it is time to eat. As the stomach fills and begins emptying into the duodenum, ghrelin levels drop. Ghrelin also activates the limbic system of the brain, which serves as the pleasure and reward center. Dieting with a caloric deficit can cause ghrelin levels to increase, making it even more difficult for an individual to lose weight. Leptin derives from adipose tissue and signals satiety to the brain. Low leptin levels activate orexigenic effectors in the brain and drive an individual to eat. Leptin levels rise when we eat and increase the activity of anorexigenic effectors to indicate fullness. In addition to leptin, there are several GI peptides involved in signaling satiety. CCK, which is secreted by the duodenum, and peptide YY, which is secreted further along the intestine, both work to inhibit secretion of neuropeptide Y in the hypothalamus, which slows eating and signals satiety. Insulin is also involved in the transition from hunger to satiety and plays an important role in regulating metabolism. Insulin secretion is at its lowest right before a meal, indicating a negative correlation with ghrelin levels. Like leptin, insulin levels rise after eating. Increased blood glucose levels following a meal lead insulin to help disperse glucose to cells, resulting in reduced levels for both insulin and blood glucose, which stimulates hunger once again.